Hi, my name is Ferdinando Almeida. I'm a PhD student at the Stein Monogastric Laboratory at the University of Illinois, and today I'm here to discuss about an experiment in which we determine the digestibility of amino acids in corn co-products, and bakery meal fed to growing pigs. Our outline for this presentation will be a short introduction followed by materials and methods, results, conclusions, and some implications of this research. In the feed industry, there is a need to explore alternative feed sources because of the increasing costs of traditional feedstuffs. Many of these feedstuffs are byproducts of the corn milling and fermentation industries or from other industries such as the baking and cereal industries. When looking at the milling industry, we have two different processes. First, the dry milling, in which hominy feed is obtained as a byproduct of the corn grits production. Distillers dried grains with solubles, or DDGS, are also produced by the dry milling process. Second, we have the wet milling processes in which corn gluten meal, corn gluten feed, and corn germ meal are produced. Corn germ meal, however, can also be produced by the dry milling process, but the one we used in this experiment was produced by the wet milling process. Let's now take a closer look at the wet milling process. In this process, corn is cleaned and steeped, and it may then undergo germ extraction, resulting in production of corn oil for human consumption or corn germ meal for animal feed. The cleaned and steeped corn may also undergo milling and washing to obtain bran and a bran-free product. The bran is further processed into corn gluten feed and used for animal feeding. The bran-free product undergoes centrifugation to separate gluten and starch. The gluten portion is processed into corn gluten meal and sold for animal feed. Bakery meal, which is another alternative feed ingredient evaluated in this experiment, is produced by mixing, grinding, and drying a combination of available commodities such as wheat products, pasta, potato chip waste, cakes, and breakfast cereals. There has been considerable amount of research to determine both the apparent or AID and the standardized or SID ileal amino acid digestibility in both corn and DDGS. However, limited research has been done to determine the AID and SID of amino acids in corn co-products and in bakery meal. And that leads us to our objectives, which were to determine both AID and SID of amino acids in corn gluten meal, corn gluten feed, corn germ meal, bakery meal, hominy feed, and to compare these values with the values for the AID and SID of amino acids in corn and DDGS. Now, looking at the materials and methods, let me first set up this graph. On the y-axis, we have the nutritional composition of these ingredients in percentage, and for this first one, we're looking at crude protein concentration. On the x-axis, we have our test ingredients. So starting from the left, we have corn, DDGS, bakery meal, corn gluten meal, corn gluten feed, corn germ meal, and hominy feed. And as we observe in this first graph, we can see that corn gluten meal is the one ingredient here that contains the greatest concentration of crude protein, compared with 25% in DDGS, 23% in corn gluten feed, and also 25% in corn germ meal. Now, looking at the NDF concentration, we observed that corn germ meal is the one that has the highest concentration of NDF at 49% versus 35% in DDGS and 31% in corn gluten feed. For the starch concentration, corn is the one that contains the highest concentration at 67%, compared with 60% in hominy feed and followed by 41% in bakery meal. For the dietary treatments used in this experiment, we formulated 8 diets 7 diets based on each of the test ingredients. So we had a corn diet with 95.4% corn, a DDGS diet with 50% DDGS and 32.9% cornstarch, bakery meal diet with 97.4% bakery meal, corn gluten meal diet with 40% corn gluten meal and 38.3% of cornstarch, a corn gluten feed diet with 50% corn gluten feed and 28.3% cornstarch, corn germ meal diet with 60% corn germ meal and 18.4% cornstarch, and hominy feed diet with 88.5% hominy feed. We also formulated a nitrogen-free diet, which was used to determine the basal endogenous loss of amino acids and crude protein. In this experiment, we used 8 pigs with an initial body weight of 82.5 kg, and they were allotted to a 8x8 Latin square design with 8 diets and 8 periods. Each period consisted of a 5-day adaptation followed by a 2-day of collection of ileal digesta. So these pigs, they were fitted with a thick cannula in the distal ileum, and digesta was collected from 8 in the morning until 4 in the afternoon on the 2 days of collection. For the statistical analysis, we used the proc mixed procedure of SAS, and we have diet as a fixed effect, pig and period as random effects. However, we did not find significance for pig and period, and therefore they were removed from the final model. For the mean values, we used the LS means procedure, and we used the PDF option if there was significance among the means with an alpha level of 0.05. Now let's look into the results, and I'll set up this graph. I'm going to show the standardized ileal digestibility only for the sake of this presentation, and we'll be looking at the digestibility of the first 4 limiting amino acids: lysine, methionine, threonine, and tryptophan. So on the y-axis here, we have digestibility of lysine in percent, and on the x-axis, we have our ingredients. So as we observed from this first graph, the SID of lysine was greater in corn gluten meal compared with DDGS, bakery meal, corn germ meal, and hominy feed, but that was not different from the SID of lysine in corn and corn gluten feed. Now looking at the results for methionine, we observed that for corn gluten meal, the SID of methionine was also greater than the SID of methionine in DDGS, bakery meal, corn gluten feed, corn germ meal, and hominy feed, but not different from the SID of methionine in corn. Looking at SID of threonine, once again, corn gluten meal had the greatest SID of threonine compared with all other ingredients except for corn and for corn gluten feed. And looking now at the SID of tryptophan, corn gluten meal had the greatest SID of tryptophan compared with corn, DDGS, bakery meal, corn germ meal, and hominy feed, but the SID of tryptophan in corn gluten meal was not different from the SID of tryptophan in corn gluten feed. Now we take a look at the SID for the mean indispensable amino acids, and we observed that the SID of indispensable amino acids in corn gluten meal was greater than the SID of mean indispensable amino acids in DDGS, bakery meal, corn gluten feed, corn germ meal, and hominy feed, but not different from the SID of indispensable amino acids in corn. So we conclude from this research that bakery meal is a poor source of digestible amino acid. Corn gluten meal for most amino acid had SID values that were greater than in DDGS and other co-products, but similar to the values obtained for corn. And the digestibility of most amino acids in corn gluten feed and in corn germ meal were not different from the digestibility of amino acids in DDGS. For hominy feed, the SID values for several amino acids were less than in corn, but because of the high starch concentration of this ingredient, it is possible that it may be used not as a protein source but as an energy source for pigs. Some of the implications of our research is that we need further research to investigate the inclusion level in the diets of each of these ingredients that were tested here. For hominy feed, there is very limited data, so we need more research in evaluating this product. And for bakery meal, we hypothesize that the low lysine digestibility may be a cause of overheating when this product is produced. Financial support for this research from Evonik Industries, the Mashoffs, and JBS United is appreciated, and the donation of corn germ meal, corn gluten meal, and corn gluten feed by ADM is also acknowledged. With that, I would like to thank you for your time, and I hope you enjoyed this presentation, and I would encourage you to visit our website at nutrition.nsci.illinois.edu, where you will find useful information on a variety of other feed ingredients. Thank you.